Welcome to the class on neuronal dynamics. Neuronal dynamics is part of the larger field of computational neuroscience. Computational neuroscience works on a theoretical description of the brain. Our brain is organized in different areas. There's an area involved in visual information processing called the visual cortex. Imagine that you're riding a bicycle and in a certain distance you see a light switching from red to green. The information about the change of light color will enter through your eye and go to visual cortex. Now you have to decide whether you want to speed up or whether you say, well, it's not worth doing it, I'm not going to make it, and you continue just as normal. While you make your decision, different areas of the brain are involved. Finally, you decide, I want to speed up, your motor cortex sends out a command and your legs move faster. The description on the level of different brain areas is a rather coarse description. In this class, we will take a different approach. We will focus on one small cubic millimeter, say, of brain area. Now, in this little area of a cubic millimeter, you will find 10,000 cells or so. And these cells form an integrated network. The cells involved in information processing in the brain are called the neurons. Neurons have different parts. There's a part called the dendrite. This is where information from other neurons will arrive. The soma is where the neuron decides whether it's going to send out a signal or not, and then this signal travels along the axon to other neurons. Each neuron makes contacts to thousands of other neurons. The contact points are called the synapses. So the axon will branch out and make different ramifications, and therefore we have not just the cells with their cell body, but we have a lot of wire. In fact, in each small area of, or small volume of 1 millimeter cube, we will have about 10,000 neurons and several kilometers of wires. What you see here on the left-hand side is a drawing of Ramon y Cajal, who did this kind of drawing over 100 years ago. And this gives a glimpse of the integrated network that those neurons form. However, it's only a glimpse, and it's still a simplification, because wherever you see empty space here, in fact, there would be other cells, there would be other neurons, there would be more material. In the end, the brain consists of a fairly densely packed material. Neurons, synapses, wires is everywhere. Now, I mentioned already that these neurons send out signals. The signal consists of a short electrical pulse called an action potential or spike. An action potential has a duration of a few milliseconds and it has an amplitude of about 100 millivolt. It's an electrical signal, it's an electrical pulse. One may ask the question how this kind of pulse, how this kind of electrical signal is generated. Just as any other cell, neurons are surrounded by a cell membrane. In the cell membrane, you will find pores. These pores can open and close. And while the pores are open, ions can flow in and out of the cell. These ions carry an electrical charge and contribute to the generation of this electrical pulse of the action potential. Next week, we will develop models, I will talk about models of this biophysical interaction of ions, molecules, and these models are called Hodgkin-Huxley type models or biophysical models, referring to Hodgkin-Huxley, who got a Nobel Prize for developing the first mathematical description on the level of molecules of the neuronal signals. Now, these models are rather complicated. They describe the biophysics. In the week after week 2, we will try to reduce the complexity of the description and we'll talk about simplified model types. Now, a particularly simple model is a model where we say, well, we don't really care about the biophysical mechanisms that contribute to the formation of an action potential. We just describe the action potential as a pulse, a standard pulse. So suppose a pulse arrives from a presynaptic neuron, from a sending neuron. It goes over the synapse, and it causes a response on the postsynaptic side, on the side of the receiving neuron. The postsynaptic neuron is the neuron receiving the signal that goes across the contact point, across the synapse. So this input pulse here will cause this response over there. Now this neuron will receive not just one input, but it will receive input pulses from many different neurons. Suppose many different pulses arrive, then the effects of all these different pulses will be added up. So this describes how different incoming pulses are transformed into a response of the receiving cell. This response is called the postsynaptic potential. But this does not yet tell us what makes this cell here decide whether it will send out a signal itself or not. Now, the formation of a pulse is triggered in this model if this membrane potential, if this postsynaptic potential reaches a value theta, a threshold value. At this point, a spike is triggered, an action potential is sent out, and afterwards we have some reset of the membrane potential to a lower value. So in this kind of model, which are called integrated fire model, in this kind of models, spikes are formal events. They are triggered at the moment when the membrane potential crosses the threshold, and then a spike is sent out, and after the spike there's some reset and refractoriness. A simple version of this model will be the topic for today, and then we'll come back to this model towards the end of this series of lectures. Suppose now we have this kind of model, and our neuron receives many spikes from other neurons. To make things simple, we assume that spike arrival is random. As a result of this, the potential will fluctuate as a function of time, and only occasionally a spike is sent out. So spikes are rather rare events triggered at a threshold. Below the threshold, in the regime which is called subthreshold regime, what we observe under random spike arrival is a fluctuating membrane potential. And this is reminiscent of what one finds if neuronal activity is recorded with an electrode in an awake mouse, for example. You see these fluctuations of the membrane potential, and spikes are rare events. From time to time, a spike is sent out. So, in lectures in week 5 and 6, we'll ask questions. What do these fluctuations mean? Is this noise? What's the meaning of these action potentials? What's the information transmitted by the neuron? What is the neural code? So overall, I will present this week a first simplified model of a neuron. Then in the next week, we will go on to Hodgkin-Huxley models, which we then simplify and reduce to what's called a Fitz-Nagumo model. We will talk about the dendrite, we will talk about neural coding, we'll talk about the question of noise, about the question of what is the information processed and transmitted by a neuron. And in week 7, in the final lecture, I will put these different things together and show how to extract from neuronal data a model of a neuron which is able to describe neural information processing, to transform incoming signal into output spikes, and therefore shed light on the questions of coding and decoding. After this short introduction, I would now like you to do a first little exercise. Please take 2 or 3 minutes and answer the following questions, and then I will continue with a first model.